step one. All right, I want to thank the presenters for inviting me to talk to you today about a story about how transcription inhibition suppresses nuclear blebbing and rupture independent of nuclear rigidity. So the real question that we're interested in is that abnormal nuclear structure is a hallmark of human disease. We've seen this since the advent of the microscope. You could look at diseased tissue and you would find the nuclei abnormally shaped. And if you look at healthy tissue, you'd see these nice elliptical or circular nuclei. And so this is true across the human disease spectrum, as you can see many of these listed here. We then fast forward in time from the late 1800s when you developed the microscope to using it for diagnostic measures via the pap smear, which was invented in 1930s at NYU, and that looked at cervical cancer cells. If they were large and abnormally shaped, that individual had cancer. How large and how abnormally shaped told you how aggressive the cancer was. Fast forward even more to now, 70 or 80 years of diagnosing and prognosing and helping cancer patients and other patients, and we still don't understand the causes and consequences of abnormal nuclear morphology. And what's also really cool here is that you can see that there's different types oh wow, fantastic, okay. So there's different types of abnormal nuclear morphology. The one we're really interested in is these nuclear blebs and this aging disease, Hutchinson-Gilford-Progeria syndrome. And here also these increases in nuclear blebbing correlate with disease progression in prostate cancer. And so why should you care about abnormal nuclear morphology and blebbing? You should care because when these form, it creates an area of high curvature and through Laplace pressure likely ruptures the nuclear envelope, causing spilling of nuclear contents into the cytoplasm and cytoplasmic contents into the nucleus. This causes cellular dysfunction. So not only is this a sort of a flag for disease, it's aiding in causing the disease state through dysfunction. Specifically, and it alters transcription and DNA damage and cell cycle control because you're spilling things from the nucleus into the cytoplasm and vice versa. This field really hit a barrier back in the 2016-17 era because perturbations of the two major mechanical components, chromatin and lamins, both cause this effect. But it had been really difficult to separate out what their different contributions were. So we developed in my lab a novel nucleus force micromanipulation, isolation, and force measurement technique of nuclei. Here you can see a nucleus being isolated from a live cell and suspended between two micropipettes. On the right, once we have that, then we can extend the nucleus to create, pull on the nucleus to create extension, and then we can watch the deflection of a force pipette with a pre-measured bending constant to back calculate force. And this allows us to get this force versus extension curve as shown here. And what we did for the first time was separate the roles of chromatin and lamins in nuclear mechanics. And what we found was that chromatin dominates this short extension force response that goes up to about 30% strain. And at that, it changes into a new linear slope because of strain stiffening due to adding on of lamin contributions. This agrees with its physiological purposes. All of our nuclei have chromatin, and so it's there to resist basic or small forces. However, lamins are not in all of our nuclei. Lamins only come on at differentiation. Specifically, lamin A is the major mechanical component of the nuclear lamina. And then lamin A is highly upregulated in mechanically demanding environments to protect your nucleus from those mechanically demanding environments. Here's the core data that backs that up. We can use a drug, histone deacetylase inhibitor called VPA to increase euchromatin, and our short extension force response decreases by about 1/3. But our strain stiffening remains. However, when we deplete lamins, we don't change the short extension force response, but we do change the strain stiffening or long extension, and in fact it goes from a strain stiffening to a strain thinning behavior. Okay, so if there's one thing I could get you to walk away from this talk, it's a sort of physical picture of what the contribution of lamins and chromatin are. And this has been backed up by rigorous physical modeling from my friend and colleague Ed Bannigan. And what you should think about for the lamina is it's like a rubber band. It's really easy to bend. The lamins have a very short persistence length, which makes them really easy to bend, but they're very hard to stretch, right? If you take chromatin, on the other hand, and you represent it as paper, it's a 3D polymer. So when you pull on it, it's immediately going to respond and resist that, right? And so when you pull the paper, and then at some point you're going to start to stretch the lamina, that's not going to come on until much later. Basic office supply model of nuclear mechanics. Okay, so once we had established these separate roles, many in the field had thought that lamins were a major component of determining nuclear morphology, and in fact we showed that chromatin histone modification state also is a major player in this game, and that is because heterochromatin provides a stiff nucleus that will resist forces and maintain shape, and that when we turn it, when it becomes weaker, decompact, more euchromatic, it then succumbs to antagonistic forces. We then went on to dictate things like the cell can sense its environment and dial up its heterochromatin to protect its nucleus. And then also, this will be important for later in the talk, but what's also important are crosslinkers. A specific crosslinker is HP1 alpha, which is also a mechanical component. And cross-linking, so your high C maps, all of those chromosome-chromosome interactions are important for the mechanics and stability of the nucleus. So what is this antagonistic force that's pushing on the nucleus? It's actin, right? So I know we're all interested in the nucleus, but actin might be new for some of you. So if you take your hand and you ball it up and you take your other hand and you place it on top of it, the top hand is actin. And it presses down on the top of the nucleus and creates confinement. And then myosin motors walking along that creates contraction, which will then squeeze that hand on top of the nucleus. And those two things are antagonistic, so in a nucleus that's weakened by perturbations to chromatin or lamins, it will then herniate and form a nuclear bleb and rupture. That's the classical view of how you get a nuclear bleb. And what I'm going to talk to you today is about how transcription has a physical role. So where's the physical role of transcription come in at, and what's the data for it? It's this. Recently, over the last few years, we found that chromatin behaves with coherent micron scale motion. That means micron scale 1 to 3 microns move in the same direction, right? And this is largely dependent on transcription. Now, if you inhibit transcription, this goes down. This has been seen by Saban's lab, which is up here, Bystrzycki's lab, Zadowska's lab was showing some work from this earlier, and Pierre Vidi. So there's a lot of labs, and I'm sure there's more that I'm not even mentioning, that are starting to find this. I should also clarify there's been a lot of MSD tracking in this conference. That's tracking a single loci and its random walk behavior, or a single nucleosome and its movement. Coherent chromatin motion on the micron scale is a much different phenomenon. So, right. So when I say transcription, what I'm really talking about is not DNA to RNA, which I've been yelled at consistently by my cell biology colleagues, but in fact the activity of transcription, which is RNA Pol II is one of the strongest motors on the cell, and it's doing a mime act where it's pulling itself along a piece of chromatin, so it's pulling in and it's pushing out, right? And so that drives coherent chromatin motions. When we did an initial simulation model with Jen Swartz's group, that group did that by themselves, not in collaboration with us, showed shape fluctuations. And then there's also disease implications. In prostate cancer, you get the prostate cancer reacts to testosterone. Testosterone activates the androgen receptor. The androgen receptor turns on a subset of genes, and that induces more nuclear blebbing. So there's a disease physiological relevance here. Okay, enough talking, let's go to the data. This is just a slide showing that we're using a very classically known drug called alpha-amanitin that binds to and degrades RNA Pol II, and so you're going to get less transcription, less RNA, and you're going to decrease your active levels of RNA Pol II. This is sort of just proof of principle. And when we did this, we saw something very crazy, which is when we inhibit transcription, we completely lose this effect of increased nuclear blebbing. So remember, this is our untreated. This is a chromatin decompacted, which then makes the nucleus weaker. It succumbs to these antagonistic forces and forms a nuclear bleb. When we inhibit transcription, it rescues this behavior. It does so in multiple cell types. Here I'm showing you 2. We've done up to about 5 cell types at this point. You can inhibit transcription at any point you want. So here's degradation. Using triptolide, you can stop initiation. Using flavopiridol, you can stop elongation. Most of these are overnight because we're lazy. And then you can use actinomycin D to actually stop transcription in about an hour and a half, and it does the same thing. Of course, you could say maybe this is just your perturbation. So we have our increased euchromatin. DSNP causes decreased heterochromatin because it's a methyltransferase inhibitor. Causes the same effect. Loss of lamin B1, knockdown of lamin A. These are all things that cause increased levels of nuclear blebbing that are completely dependent on transcriptional activity. Okay, so to prove that we have a new novel mechanism, we need to go through our classic mechanism and prove that none of these are changing. So we looked at chromatin compaction, and in fact, in our VPA, the idea is that it's going to increase chromatin. Actually, with alpha-amanitin, it makes it even worse. Heterochromatin doesn't change, so nothing's changing there. We can use our force measurement technique. Remember, that can separate out the role of chromatin via the short extension, and that decreased mechanics stays. This nucleus is still weaker, but is failing to form a bleb. So that's not it. Then we looked at lamins, and because we can do this strain stiffening, we can see that the lamin contribution to nuclear mechanics does not change. Lamin levels do not change. What's interesting, though, I'll note, and some of you are interested in this, is we do see these lamin invaginations. And the idea here is if transcription is causing coherent motion to push against the periphery of the nucleus, if you have less of this, maybe the nuclear envelope is less taut. Then we asked if we're changing our antagonistic forces, and this would include actin confinement. Because our nucleus is weaker, because of chromatin decompaction, you can actually see that decrease, and that decrease has remained when we treat with alpha-amanitin, so it's not changing actin confinement. And then actin contraction can be measured by the number of phosphorylated myosin motors, and that does not change either. And here's a positive control, which is a Rho inhibitor, in which you would see change. So here's proof that the classic mechanism is insufficient to explain nuclear blebbing, and thus we found a new novel component, which is internal transcription-driven chromatin motion. At least this is what we are currently hypothesizing. So these RNA pol motors are pushing the chromatin. This coherent chromatin motion is pushing against the periphery and bulging out to form that nuclear bleb. If this was true, then it would be harder to form a bleb, and maybe those blebs would be less stable. And that's, in fact, exactly what we see. So at the top is sort of our classic perspective, untreated and VPA. You have a normally elliptical nucleus. When that bleb begins to form, a nuclear rupture event happens immediately after it. These nuclear rupture events are actually really efficiently repaired. So the nuclear envelope is repaired in about 10 minutes, and that NLS GFP will reincorporate into the nucleus, and when it does, that bleb will stay. Blebs are very plastic. Blebs stay for a very long time in most of our sort of normal/untreated or perturbed conditions. They can stay for the entire interphase cell cycle and require mitosis to reset nuclear shape. However, when we inhibit transcription, not only do you form less blebs overall, when you do form a bleb, we saw this brand new phenotype, which is you form your little herniation, you rupture, and in the 10 minutes it takes for you to repair your nuclear envelope, that bleb has been reabsorbed back into the nuclear body. This suggests that coherent motion is not only important to press out to form the bleb, but to stabilize that bleb. If it's true, then we might also be able to destabilize blebs that are currently happening. And so we looked, and in fact, what I told you is true. When you look over 8 hours, blebs that exist at a very low rate, this is like 3%, will reabsorb, right? So blebs are very plastic. They tend to stay. We talk about them as plastic deformations. But when we inhibit transcription with any of our transcriptional inhibitor drugs, a third of those blebs are going to be rescued back into the body. This means that active transcription is important for forming the bleb, stabilizing it after that immediate formation, and maintaining that stabilization over a long period of time. Okay, so like we like to do all of our experimental stuff and then have a simulation model. So we've built a nuclear stiffness model through Ed Bannigan, right, and our work together. We were both in John Marco's lab, which is was a great group to be in. Also, other people here. And recently we've added to that chromatin cross-links, right, via HP1 alpha and high C contacts. So what you get is this lamina in the purple. You get chromatin on the interior. You get chromatin-chromatin cross-links. That's your H1P1 alphas or your high C contacts. And you get these linkages, chromatin-lamin linkages. That would be your LBR, your nonspecific chromatin-to-lamin binding, and so on and so forth that have been mentioned at this conference. We have done less studies on that. But they're all built in here. Then you drop in a repulsive or contractile motor to represent RNA pol 2 pulling or pushing. And then you dial up number of motor activity. And when you have a low number of motors, you can stabilize your shape. But however, when you have a lot of motor activity, you get these bulges. Now, bulges in a simulation are not the same as blebs. Blebs are very complicated, but this is the simulation equivalent. So why is this bleb forming, or why is this bulge forming? When we go in and we look at the trajectories of our simulation, we can see here that coherent motion from the green and the blue are pushing out to create this bulge, right? And that nicely agrees with the simulation, or this simulation data nicely agrees with the experimental data that there are coherent chromatin motions over micron scale. Okay. What I also want to share is that story is published. You can go read it. That's why I did this talk. I also thought this talk would fit better with the things that were going on today. What you ideally would want is you would want to degrade RNA Pol II as fast as possible so that you don't mess up DNA to RNA actually reading of the genome. You also wouldn't want to change reading of the genome. Maybe you just want to slow down RNA Pol II. And then you wouldn't want to turn off transcriptional activity, but what if you could sort of dial it down and dial it back up? Well, we can use an RNA Pol II GFP AID construct to degrade RNA Pol II in 5 hours. And in fact, we get the same effect with a completely different cell line, HCT116s. I think this data is the most crazy and maybe one that will generate a lot of talk, which is you can use camptothecin, a DNA topoisomerase 1 inhibitor, which is supposed to alleviate supercoiling during transcription and is known to basically slow the physical activity of RNA Pol II walking across the DNA. And by simply slowing down transcription but not changing overall transcription, you can suppress nuclear blebs. And this is, I think, our strongest evidence that is the physical activity, not the actual DNA to RNA reading. Then you can use serum starvation, when we can do this by your normal 10% FBS, and then you can starve with lower levels, and we can add it back. So this is 30 minutes in. It would be nice if we had the 0. But this is preliminary data, so it's a little messy, apologies. You can see that when you serum starve, you suppress the ability to form nuclear blebs. And it's well known that when you add serum back, that transcription increases in the next hour or 2. And so 2 hours later, your nuclear blebbing percentages are back to where they should be, right? And right now, we're obviously getting the data to show that there's decreased levels of transcription and then increased levels of transcription when you add this back. We're also very interested in the future at looking at chromatin motion, right? So what we have here is a very sort of simplistic movie to show that there's these 2 sort of less dense spots, and they move into the nuclear bleb, but the spots that are further away move less. So you can see a lot of movement in the purple and yellow, and less movement in the ones that are further away. And we hope to sort of do coherent chromatin motion to show how important it is for nuclear bleb formation. Finally, we're also building out a story about how, what I told you earlier, which is that in prostate cancer cells, these LNCAP cells, you can add DHT, dihydroxytestosterone. This activates the androgen receptor. The androgen receptor then goes in and turns on a subset of genes. And this behavior by itself increases nuclear blebbing. And this is important because this is a Gleason score, a low Gleason score, or a normal in a human tissue, and this is an aggressive cancer in a human tissue. This difference between 2% and 8% nuclear blebbing is physiologically and disease relevant. If we add back transcription inhibitors, we block this effect. We have new data that if you add in an androgen receptor blocker, you also stop this effect, so that it's completely dependent on that. And then I want to get back to, again, wait a second, I lost track of why I should care about any of this. You should care because when you form a bleb, you cause ruptures. And you place that DNA into a new compartment, which we think is a restricted compartment, and you increase levels of DNA damage. It's well known in prostate cancer and in many others that you can get these specific translocations that drive disease. One of our future hypotheses is when you form a bleb, you are creating DNA damage, you are segregating DNA into a specific compartment, and you are making an ample opportunity for translocations to happen that drive disease. Can you guess which genes end up in the nuclear bleb? The genes that are on. All of the transcription, the AR responsive genes tend to localize to these nuclear blebs. Again, showing how important transcription is to the formation of a nuclear bleb. So the conclusion for this talk is that we have this model, which is that transcription motors can push stuff around. Actually, I missed the most important thing to this model, so mistake, but it'll highlight through failure we can learn, as I teach my students. Actually, what's really important about this is that you have cross-links between all of this chromatin that should be here in red, your HP1 alpha, your high C contacts. The reason that you get coherent motion is because it's not one RNA pol pushing and pulling, it's a set of them pushing and pulling highly cross-linked domains, micron-scale domains, of chromatin in the same direction, and that pushes against the periphery to bulge out and form and stabilize a nuclear bleb. This is a new, novel, independent of the established nuclear rigidity and actin antagonism force balance. Chromatin coherent motion is a plausible new mechanism for this formation that we're working to provide data for. And that transcription activity in disease can drive nuclear blebbing, leading to more dysfunction. There's many types of these. So I gave you testosterone, but you can think of MYC or any of your other transcriptional activators that are heavily active in diseases, and many of those diseases present abnormal nuclear morphology. Last, I want to say thank you to my collaborators at Bannigan and Jen Schwartz, who are the leads on the modeling. Sartak Gupta was the lead on the modeling specifically. Many people at Northwestern, and all my students. It's also the point in the talk where I tell you that my entire lab is run by undergraduate students. So all of the work you saw today is done by either a fifth-year master, Isabel was a rotation student from Germany who came for 6 months, and this is her paper that we're talking about, and all of these are undergraduates that provide supporting information. Thank you.
Questions? Any students want to step up to the mic? Give you guys a chance to take the first shot at questions. Postdocs invited as well? Yes.
Hi.
Hey.
Thanks, Anshu. Very nice talk. So I have 2 follow-up questions. In this model, you assume that the mechanics of the membrane is basically unperturbed. And have you actually checked that? Because in your mechanical measurements, you can actually check the contribution of the membrane if it's actually different, or is it exactly the same?
All of our mechanical measurements of the membrane, the lamina, and the chromatin show no change, no measurable change.
After transcription suppression?
Correct.
Okay. And the second question is, what about if you increase the level of transcription? Do you see increased plebs?
So that's essentially what the DHT experiment is. This experiment is, we're increasing transcription by adding a hormone that would activate the androgen receptor, which is a transcriptional activator, and that is increasing transcription. The sort of pushback we get from a lot of people is, that's only turning on a subset of genes. How can you get nuclear blebbing from turning on a subset of genes? And I say, that's a great question, but that's currently what the data is lending towards. And so I think there is a question, which is how much transcription do you need to turn on to get these blebs?
Thank you.
Hi, very nice talk. Maybe it's a naive question, but I wonder whether you can separate the action of RNA pol-2 moving, so the active process, versus just the production of RNA, just the content of.
Right.
Because I heard this beautiful talk at some point where a person was showing that there's apparently a constant in terms of a mass of the molecules in the nucleus versus cytoplasm across cell types, across species is
right.
constant, right?
Right.
So I would imagine that, just the amount of molecules in the, right? So if you have a lot of RNA, that will also push against the membrane.
Right, so the CPT, the camptothecin, is a DNA topoisomerase 1 inhibitor, and the idea is that it's supposed to slow transcription, but not change overall transcription output, right? So we're going to measure that to see if there's a significant change in RNA output.
Right, but are you measuring in this experiment how much of RNA in total RNA you have?
Yes, we have RNA measurements that you can do via this click chemistry. So we are doing those measurements to see if they do change.
All right.
I'll flip it on you, which is maybe that's too hard. So what you need to do is drive coherent motion. Now Pierre Vidi's group has shown that a RAD51 inhibitor, BO2, increases coherent chromatin motion. So we fed our cells BO2 to increase chromatin motion. We haven't done all of the experiments, but initially that thing, which is only supposed to increase chromatin motion, increases nuclear blebbing. And it does so in a way that looks like changes in transcription because the blebs form and they tend to be temporary. And so that's another direction that we're headed in.
Thank you.
Yes, over here.
So I guess this is a question coming from someone who's thought about cellular blebbing, but not so much nuclear blebbing. So in the cell, what the bleb initially is, at least, is a breaking apart of the connection between the cell membrane and the actin cortex.
Yep.
So the first question is the initiation of a bleb here the same? Is it a breaking of the connection between the lamin and the underlying chromatin structure? If so, if that's true, which I guess you're shaking your head yes, so maybe it is.
No, I think what you're outlining is a grant I want to write in 5 years. Like, I think we're working towards specifically what the nucleating event of causing a nuclear bleb is. I think there's this idea of frustration that Ed and I have talked about, where maybe this polymer is getting bigger and this polymer is not, so it gaps the second one and it pops out because it's too big to be held down, the lamina and the euchromatin. There could be loss of connections, but that takes a lot of really fine measurements to prove, right? So we're not quite there with exactly what the nucleating event
So
of a
I was
nuclear
going to
bleb
ask
is.
if you'd made any progress, essentially, in what you're saying, in seeing whether the expected levels of force you would generate by this coherent motion is really consistent with, for example, breaking some type of connecting bond between lamin and underlying chromatin structure.
I don't think so. So what happens is in our weak, I just had to skip over this for the size of the talk, but what happens in our VPA, it's still weak. So actually what happens is you still get some ruptures, and they're what we call non-bleb based ruptures. So the nucleus just tends to, under its compression of the actin, rupture, but it can't form a bleb.
Yeah, that's not a bleb. At least in cellular context, that's not a bleb. A bleb is really a region of sort of between the membrane and the underlying, let's say, elastic structure.
Right.
In the cell, the actin cortex, in the nucleus, presumably the chromatin.
Right. Yes, I think that we're working towards trying to figure out if the model you're putting forward is true or not, and we don't know that. What I'm telling you now is that if you don't form a bleb and you're weak as a nucleus, you still rupture. So I think the coherent motion is just capitalizing on this compression and then sort of moving into this empty space. So there probably, where there was going to be a rupture without a bleb, there probably would be a rupture with a bleb. That's what I'm sort of hypothesizing.
I guess the related question is, again, in the cellular context, there are blebs and then there are ruptures, and they're not exactly the same thing. I mean,
right.
One can lead to the other.
Right.
Is this true? This is presumably true here also. You can get blebs, but you need some additional thing, or at least stronger thing, to cause it eventually to rupture. Is that true?
Almost, so I would say it's basically 98% of the blebs that form rupture. So they're pretty heavily tied, but it's biology, so it's not perfect.
Okay, thanks.
I was wondering if you look at the effect of air loop accumulation.
What accumulation, sorry?
By air loop accumulation, because air loop accumulation can lead to micronuclei formation, so there is some paper showing that. Maybe when you inhibit transcription, you can accumulate air loops, and this can have an effect on what you are looking at.
That's a great question. I know very little about L loops, but we will go looking for them. What I can tell you is micronuclei form from mitotic failure, and nuclear blebs form from an ellipsoidal nucleus herniating itself, and so those are two different phenomena.
Okay, thank you.
I just want to continue a little bit on what Herbie was commenting on it. You have many phenomena going together, and you say about this transcription. You have the polymerase motors that act on the surface. You have all this cross-linking, and all these things are tied together. Do you have other motors inside that basically that are also involved on this process, or is just the only motor is Pol 2? And what are the time scales for this cross-linking compared to creating motors and acting on it?
The cross-linking is built in. It's like defined. Like there's a cross-link every so many kb.
But you increase it, right? Basically, when you start during the process, don't you?
Yes, I think that during transcription, cross-linking probably does go up and add to it, but I would argue there's probably enough cross-linking inside the genome. The work we did with Joop Dekker shows that there's essentially an interaction from chromatin to chromatin every 25 kb, so I think that's pretty...
But if you increase the cross-link, don't you change the elasticity of your chromatin, and that's going to affect what you're...
Yes, so then from the force measurements, we would say that we're not drastically changing the cross-linking from transcription to lack of transcription, because the mechanics stay this similar.
And the motor is only polymerase, or there's other motors that are really involved with the process?
The motor we write into the simulation is a naive sort of extension or contractile motor. RNA Pol2 we're investigating here, there are all obviously other motors inside of the genome. We did DNA replication. DNA replication machinery does not appear to impact nuclear blubbing. So we think it might be an RNA Pol specific thing. And then the question is, why is one doing it and the other isn't? And I think that's an interesting question we should look into.
So looking at your some of your XZ images, it looks to me like these nuclei are actually pretty flat. They're pretty compressed.
Yes.
So they're pretty close to the surface. So one of the things that we've been finding is we've been looking in prophase, and we've noticed coordinated movements of lots of chromosomes in prophase, which I don't think has ever been described before. And the reason is that's movements of the nuclear envelope. Has it occurred to you to can you put trackers on the surface of the nucleus and see, is the chromatin flow that the chromatin movements you're seeing, is that actually driven by the envelope? Which then would be a connection between movement and something that could lead to blebbing, because something would be happening in the envelope.
That's a great experiment. I think that we could definitely do that. I don't know that that was on our radar. I think some people talk about how the nuclear envelope is more static, but I agree with you that I think it can be fluid at times.
Yeah, well, particularly, so what happens in prophase going into mitosis is centrosomes are moving around and microtubules are going in. You have incredible changes in the shape, but you're in interphase.
Right.
So I don't know, and I don't know that people. Well, nuclei rotate.
Right.
There are lots of movies.
They do. They rotate a lot. Yeah.
So there could be movement.
Yeah, I think this is a good experiment. I think we will look into this for sure.
All right. With that, let's thank our speaker again.
All right, great. So first, I would like to thank the organizers for the invitation and also for making everything work online, given my kind of visa issues. So I'm currently a postdoc in the lab of Geneviève Olmosny, where I did my PhD. And today I'm going to give you kind of a brief overview of what I was doing during my project, which was trying to place the variants of Histone H3 in the context of higher order chromatin organization. So histones, together with DNA, form the basic unit of chromatin, which is the nucleosome. And then chromatin goes on to have higher order folding, as I'm sure you're aware, and you become even more aware during, during this conference. So in addition to kind of helping package DNA, nucleosomes are also important for the regulation of key processes like transcription and replication. And this can happen both through modifications or through the incorporation of different histone variants, which is what the lab is interested in and what I'm going to be talking about today. So what we're focused on is variants of the histone H3. And H3 is one of the core variants, which together with H4 forms the core of the nucleosome. So there are four kind of very well-studied variants, which are, so first H3.1, 2, and 3, which as you can see have very high homology between each other. And then SEMPE, the centromeric variant, which is actually quite divergent. So if we focus on H3.1, 2, and 3, if we actually look at the structure that they form in the nucleosome, this doesn't seem to be very different, which is in line with the fact that they have very few amino acid differences. However, so why do we need variants? If we actually go kind of beyond amino acid sequence, what we see is that they're very distinct both in their expression, but also in their deposition modes. So H3.1, which is basically functionally identical to H3.2, so I'm going to be only referencing to H3.1 here, is known as the replicative variant due to its peak of expression in S phase. And it has a particular mode of deposition, which is coupled to DNA synthesis. So this happens through its histone chaperone Cap1, which can interact with pCNA, which is part of the replisome. So in this way, it's ensured that newly synthesized DNA can be efficiently chromatinized. So this leads to basically the deposition of H3.1 all along the genome, and this happens throughout this phase. In contrast to H3.1, we have H3.3, which is the main non-replicative variant. So its expression doesn't peak in S phase, it's expressed throughout the cell cycle. And its deposition is also independent of DNA synthesis. So this can be mediated by several chaperones, but the one that I'm going to be focusing on today is the chaperone Curie. So Curie can deposit H3.3 again throughout the cell cycle, and it does it at particular places. So Curie can ensure H3.3 deposition in a manner coupled to transcription, which ensures H3.3 deposition of active genes. But it can also have this gap filling function where it can recognize naked DNA, and this ensures efficient chromatinization of places which might have lost nucleosomes for one reason or another. So what this results in is enrichment of H3.3 at active sites, nucleosome gaps, and also regulatory elements. And in addition to this, other chaperones can deposit H3.3 at the telomere and pericentric heterochromatin, while the centromeric variant SNP is found specifically at the centromere. So if we go kind of away from the schematic view and we look at some data, this is some ChIP-seq looking at the ratio between H3.3 and H3.1. And indeed, you can see how H3.3 is found enriched in these specific peaks, while H3.1 is found in these large domains. And if we compare this to genome function, what we can see is that the peaks of H3.3 enrichment largely correspond to places of nascent transcription, whereas these large domains of H3.1 largely coincide with places associated with inactive chromatin. In addition to transcription, the two variants are also correlated with replication. So you can see that H3.3 in these active regions tends to be also where early replicating chromatin is when we assay this by RepliSeq, whereas H3.1 tends to be found in these late replicating regions. So far, we've had this kind of linear map of the variants, and what I wanted to understand was how can we place this in the context of higher order chromatin architecture. So moving from basically this nucleosomal scale to higher order structures like topologically associating domains or TADs, which are these regions which tend to be enriched interactions within and associated and insulated from neighboring regions, which you can find within these megabase scale compartments, which tend to be large regions of active or inactive chromatin. So what we really wanted to understand is what is the impact of the deposition of different variants on these different scales. So to do this, we went back to H3.3 and Curie, and we used cells in which this chaperone is constitutively knocked out. So this allowed us to dissect how the specific nucleosome assembly of H3.3 in particular regions can impact higher order organization. And so to assay the effect on genome organization, I used Hi-C, which also I assume I don't really need to introduce as a method. But here I'm just showing some examples of where you can find compartments kind of away from the diagonal as displayed pattern, and where you can find TADs, which is along the diagonal in these kind of triangular domains. And what I'm going to be focusing today is mainly compartments, although we did also look at TADs. So the first question that we wanted to answer was, okay, how does the distribution of the variants correspond to where compartments are? So this is an example of Hi-C map of chromosome 5 arm Q. So you can see an example of our map, and then a track with A and B compartments go in red and blue. And below we have the enrichment of H3.3 and H3.1. And you can already see that H3.3 you tend to find in active domains, whereas H3.1 you tend to find in compartment B. And we can summarize the genome-wide by performing these heat maps, where basically each line is a compartment. So they're sorted from the smallest to the largest, and they're centered in the middle. And then they're colored by the enrichment of H3.3. So you can see that indeed genome-wide, H3.3 shows enrichment in A and a depletion in B compartments. If we look at H3.1, you see a somewhat reciprocal pattern. So we can see that H3.1 tends to be kind of depleted within A and then enriched in these large B regions. This pattern is not exactly reciprocal, and this makes sense because we know that H3.1 is deposited with replication kind of throughout the genome and only exchanged in particular places by H3.3. Whereas this enrichment in the large B regions is likely because they are the ones that show the least turnover. So these are the places where H3.1 is least likely to be. So after we had done this first characterization, we wanted to see what happens in the absence of Curie. So the first thing to check was, are compartments themselves actually affected? So this is the same map that I showed you before from the wild type, and this is the map of the same chromosome from human knockout cells with the compartment track above. And you can see that there doesn't seem to be a lot of compartment changes. And indeed, we can quantify this by doing scatter plots of the eigenvectors, which are what is used to call compartments, either from wild type or knockout cells. And you can see that we detect significant but small proportion of the genome with changes compartment. So it seems that largely compartment identity is maintained in the absence of Curie. So this allowed us to then look at what happens to the variant distribution. And here I'm going to focus on H3.3. So you can see that in Curie knockout, we have this decrease of H3.3 enrichment throughout compartment A, with an increase in the enrichment only in these large B regions. And in addition to this, we could see that the enrichment of H3.3 at the boundaries became kind of a bit more fuzzy. And we also looked at H3.1, but we didn't see any corresponding change. So after seeing this distribution, we wondered, okay, this is not affecting compartment identity, but is it actually affecting the way that compartments can interact with each other? So to do this, as a first look, I computed these differential maps at 1 megabase resolution. So this is the same chromosome that I was showing you before, with the eigenvector track below, which can also show you that there doesn't seem to be much compartment switching. So first, if we look at, for example, two-way compartments that you can detect here, we see that they seem to have decreased interactions away from the diagonal, whereas if we look at these large compartment B regions, this doesn't seem to be the case, and if anything, they seem to interact slightly more. So again, to kind of look at the genome-wide, what I did were saddle plot analysis, where basically you take the entire high C map, and then you resort it based on the eigenvector value, going from the lowest to the highest. So I did this in the wild type and the knockout, and this is the differential map of the two. So you can see that indeed we can see kind of reduced interactions here, and also kind of in the differential more clearly in A with A regions, but then also kind of A compartments with the rest of the genome. So to quantify this, I stratified this saddle in 6 areas. And quantifying it, indeed, I could see that there was a decrease of A interacting with itself, with B, or with this kind of intermediate regions, whereas B seemed to increase contact only with these kind of more intermediate regions, most clearly. And as I said, we also looked at pads, but we didn't detect any changes in the position of the boundaries or their local isolation. So this effect that we could see of the variant was really on the scale of compartments. So the next question was, okay, well then how is Curie and then potentially H3.3 doing this? So the first thing that we thought to look at was histone PTMs, because on the one hand, PTMs have been put forward as potentially important for compartmentalization, and on the other hand, the two variants have been associated with different PTMs. So we profiled them by native chip, and we selected a set of active and inactive marks. So we took K4 mono and trimethylation as well as K27 acetylation as examples of active marks. And indeed, you can see them enriched in compartment A with H3.3 here as reference. And we also looked at K9 and K27 trimethylation as examples of inactive marks, which we could see enriched in compartment B. When we looked at the Curie knockout, again, we could see this redistribution of H3.3 from A to these large B regions, but this was not matched by a redistribution of the PTMs at the scale. So it seems like the effect that we're seeing is not through an effect on PTM distribution. So the next thing we wondered was, okay, well, are actually impacting the deposition of histones, so maybe this is impacting the density of nucleosomes and chromatin accessibility. So to look at this, we performed the TAC-seq, and again, this is data within A and B compartments, so you can see that in the wild type, we tend to have accessibility in A and not so much in B compartment, similar to what we see for H3.3. In the Curie knockout, strikingly, we see that accessibility seems to be increased in A while it decreases in B compartment, along with this redistribution of H3.3 enrichment from A to large B regions. So this analysis, we did 10 kb, which is quite large scale. So then we wanted to go kind of to the nucleosomal scale and understand what's going on. So the first thing to do was to actually look at the TAC-seq peaks. So I did peak calling, and there I could identify a similar number of peaks from the wild type and the knockout with very large correspondence to each other. So this distribution didn't seem to be because of a change in where peaks are. However, when we looked at the attack signal at these, what we could see was that there seemed to be a decrease upon Curie knockout. And when we compared the two variants, what we could see was that in the wild type, attack peaks were enriched in H3.3, as we would expect, and this was strongly decreased upon the loss of Curie, whereas H3.3, which was normally depleted at attack peaks, tended to increase, indicating that there might be some compensation. So potentially in the wild type, you would have Curie targeting H3.3 there and promoting dynamics, and in its absence, you basically don't replace the H3.1 that you originally put there. However, this was not really the effect that we were seeing on the scale of compartments, so then we wondered, okay, where could this be coming from? And like I mentioned already, Curie has been put forward as important to deposit H3.3 in the context of. So because of this, we looked at active genes, and here I'm going to focus on the top 5%, but we looked kind of stratified by expression level. So the first thing that we did was we also had RNA-seq data, so we checked that we didn't have differential expression of these genes, so the changes that we could see were not due to changes in their expression. So if we look at the top 5% expressed genes, what we see in the wild type is that they have a peak of accessibility at their TSS, but then the gene bodies are not really accessible. And this is accompanied by an enrichment of H3.3 throughout them and H3.1, which is not particularly enriched there. If we look at the Curie knockout, what we can see is that in terms of attack-seq, we have this decrease of accessibility at the peak, which is similar to what we see at the attack-seq peaks. And this, again, is together with decrease of H3.3 and an increase in H3.1 enrichment. But when we looked at the gene body, what we could see was that actually attack-seq signal increased throughout the gene bodies, again, together with the decrease of H3.3 enrichment, indicating that Curie kind of has two roles. What active gene promoters, it can promote potentially dynamics, H3.3 deposition and accessibility, whereas throughout the gene bodies, it's important to maintain nucleosome density by both depositing new H3.3 and recycling in the context of transcription. So we saw this and then we wondered, okay, well, could we see actually any change in the organization specifically of active genes? So for this, I did some pileups along the diagonal to look at their local organization. So these are centered at the TSS, and then I'm looking at half a megabase flanking upstream and downstream. And you can see already from the pileups and then clearly in the differential that there seems to be a reduction in this kind of long-range insulation, while there seems to be also a decrease in the local interaction within active regions. So to put all of this together, what we try to do is a summary of how we think Curie may be impacting chromatin organization across scales. So what we already knew was that Curie is important to enrich H3.3, to deposit H3.3 at active transcription start sites, at regulatory sites, and also in the gene body. And what we could see now was that in its absence, active promoters and regulatory sites decreased their accessibility and seem to gain H3.1 enrichment, whereas active gene bodies, on the other hand, end up with higher accessibility and potentially lower nucleosome density. When we zoom out and look at how this impacts the kind of 3D organization of local genes, we can see that it's both reducing insulation and at the TSS, and it results in lower interactions locally, while it doesn't seem to be impacting TAS on the whole. And when we zoom out even further, what this translates to on the level of compartments is that in A compartments, which would be containing active genes, we see higher accessibility, reduction of H3.3, and reduction of contacts of A with all other genomic regions without a redistribution in PTMs. So in addition to kind of placing the variants in this context of higher order organization, what we also wondered was how can this be related to another role of Curie, which the lab had recently shown, which was in defining early initiation zones. So to give you a brief introduction, a study from several years ago in the lab was looking at the de novo deposition of the two variants, specifically in S phase. And what they could see was that while H3.1 deposition basically travels with the replication fork, H3.3 is continuously targeted at regions of previous enrichment. And what they could identify then was the sites of H3.3 enrichment flanked by H3.1, which mark the boundaries of early initiation replication. So then the question was, okay, well, is this just because of this link of the variant deposition with the replication or the independence of H3.3, or is this functionally important? And here is where they use the Curie knockout to actually identify two types of sites. So they could see that in transcribed regions, in the absence of Curie, these sharply enriched H3.3 sites would become blurred, and the replication of the boundaries would become fuzzy without impacting their expression. While at another subset of sites, which they call buried, they lost completely the H3.3 enrichment and this early replication. And this was at sites which tended to be lower non-transcribed. So it seems that Curie was important to define early initiation zones in a manner which was independent of transcription. And this is just to show you kind of what they look like. So these are, again, a set of blurred and these buried sites. So these are the same type of heat maps that I was showing you before, where this is colored by the signal of H3.3. And you can see that in the wild type, they're both H3.3 enriched. While in the Curie knockout, we see this blurring and reduction of H3.3 enrichment at the blurred sites and this complete loss of H3.3 enrichment and the buried sites. To profile early replication, what they did was look at EGU incorporation at 2 hours in S phase. And again, you can see that in the wild type, these sites were early initiating both the blurred and the buried, whereas the knockout, you can see this blurring at the blurred sites and this loss of early initiation of the buried sites. So what we wondered was, okay, well, how is Curie doing this? Because again, early initiation zones have been put forward to be enriched in active marks, so active PTMs have been potentially linked to them, and they have also been shown to be accessible. But then there was also potentially a 3D component to this, as early replication strongly correlates with the presence of, or with being in compartment A, and also early firing origins are more often found at the tab borders. So given the fact that we saw an effect of Curie on this hierarchy organization, we then wondered, is this also important for its definition of early initiation zones? So to look at this, we actually looked at a lot of things, but I'm only going to show you the 3D organization part, but I'm happy to also talk about the rest. So to understand what's happening to their compartment organization, I did the same type of scatter plot analysis that I showed you before. So here in gray, you have all genomic bins. In green, you have the blurred sites, which again had fuzzy early initiation, but they were still early replicating. So we see that the majority of them are found in compartment A in the wild type, and basically this remains the same in the knockout. Whereas if you look at the buried sites, which were early initiating the wild type, but they lost this in the knockout, we could see that actually already in the wild type, there was a proportion of them that we could find in compartment B, indicating that you can have early initiation in the compartment, kind of starting to separate the two. And when we looked at what's happening upon the Curie knockout, we compare the distribution of sites which changed or remained in the same compartment to a set of random sites which were matched for size. So we can see that the blurred sites don't seem to significantly switch, whereas for the buried sites, almost a quarter of them switch compartment, and the majority of the switching actually goes from A to B, which, considering the loss of Curie, would kind of make sense. But this was only partial. So even though all of them lost early initiation and were not highly transcribing to begin with, only some of them switched compartment, again, kind of indicating that there might be a separation between the two. So to try and understand if maybe the compartment identity of these particular sites was important, what we did was perform a Curie rescue experiment. So we supplied Curie back, and we wanted to see if we could regain both the H3.3 enrichment, the early replication, and this compartment identity. So I'm showing you the same heat maps that I introduced you before. So here we have the wild type and the knockout rescued with 5 PS control, where you can see this reduction in blurring of H3.3. And you can see that upon Curie rescue, we can get reduction of the blurring and the sharp H3.3 enrichment on these sites. And this is summarized here. If you look at the buried sites, again, you have this loss in the knockout and in the Wkshp rescue, which again is restored upon Curie rescue, indicating that we can recover enrichment at both types of sites. We then looked at early replication, and again, you see this blurring in the knockout rescue to Wkshp, which is reduced upon Curie rescue at the blurred sites, and at the buried sites, you see this loss of early initiation, which was then regained in rescue with Curie. So we could recover both H3.3 and early initiation. So then the question was what's happening to compartments. So again, we performed Chi-C in the Curie knockout rescue to Wkshp or with Curie. And here from the scatter plot, you can already see that the sites don't really seem to be switching. And indeed, when we quantify this, we detect much lower levels of compartment switching, which is nevertheless significant for the buried. And the majority of it is from B to A compartment, so the reverse of what we observed originally. However, it doesn't seem to be to the same extent, so it looks like the rescue can increase the H3.3 enrichment and early firing at the buried sites without large reversal of B to A changes. And indeed, when we compared the enrichment of H2.3 and the early firing at either sites which switched from B to A upon rescue, or the ones that remained in B, we could see that both types of sites could rescue, and we didn't see a significant improvement of rescue of the ones which switched from B to A compared to the ones which stayed. And this is just a representative from some sites which switched from A to B and then remained there, or some sites which were always in B. So here I'm showing you the ratio of H2.3 to H2.1, enrichment of H2.3 and early initiation. And you can see that the sites were H2.3 rich in the wild type, which was gone in the knockout, and then we start regaining this in the rescue. And indeed, we also see an increase of the EDU signal. And importantly, we only see an increase of EDU where we also see an increase of H2.3. So for example, here you have two sites which are not lower rescued, and here we also don't see an increase of EDU. So in summary, from kind of the two stories that I told you, summary from this part first. What we see when we look at how H2.3 defines early initiation, we started with these two types of sites, the blurred and the buried sites. So we could see that blurred sites tend to be in compartment A, in line with what people think of early initiation replication zones. But when we look at the buried sites already in the wild type, we can see that there are some both in A and in B compartment. When we look at what's happening in the Curie knockout, for the blurred sites first, we can see that they remain in compartment A. And upon Curie rescue, we can recover the precision of H3.3 enrichment and of early initiation while they again remain in A compartment, indicating that this precision is not related to their compartment identity. Looking at the buried sites, which were more strongly affected by the Curie knockout, what we see is that in the absence of Curie, there's a proportion of them that switches from A to B with the loss of early initiation, but not all of them, again indicating the two can be disconnected. And when we rescue with Curie, we can again regain H2.3 enrichment and early initiation without a substantial switch from B to A compartment, indicating that Curie can define these early initiation zones independently of their compartment. So now for summary of the two parts. What we have looked at is how this Curie histone deposition pathway, which is independent of DNA synthesis, can have two important roles. Looking at this multi-scale chromatin organization, what we could see was that Curie is important for the nucleosome density in active regions linked to transcription, and this results in weakened interactions in compartment A on a genome-wide level. Whereas when we look at the early replication initiation zones, which is a function of Curie not related to transcription, we can see this is also irrespective of the compartment of these early initiation zones, where we can influence both the precision of early initiation at blurred sites and the detection of early initiation at buried sites without an absolute correlation to their compartment. So, thank you very much for listening. I also want to thank very much, again, the organizer for inviting me, Geoff for taking me in, and for supervising me during this work, and Audrey and Jabir, and Alberto, a of the lab, who also helped with the experiment, the analysis, and the rest of the team for the support. And I'm happy to take questions.
We have time for a few questions. And from Sasha, yeah.
Yeah, hi Tina, thanks. I had a question about CTCFs, because we know that CTCF is well positioning nucleosomes around it when it binds. And my question is, like, when are these nucleosomes deposited, and do you see any effect in area knockout?
I have some supplement for you. So let me find this. All right. So we looked globally at CTCFs, and we didn't really see a difference in terms of pileups. This is then CTCF sites, which I segregated by association with the transcription start sites. So either some which are far, so more than 50 kb from K4 trimethyl peak and not in gene bodies, and some which are near transcription start sites, so they overlap with K4 trimethyl peak. And you can see that the ones which are far, they have some reduction in accessibility, though not much. Indeed, Hp.3 has been linked with CTCF sites, and we do see a reduction of Hp.3 enrichment at CTCF sites. But when you look at the ones which are far from the TSS, you basically don't really see an effect on insulation. But if you look at the ones which are near the TSS, this is where you see the strong reduction of accessibility, and also where you see this reduction of long-range insulation. But if you look, what I think is interesting is if you look at really the corner where you would expect the site to be, you don't see such a reduction. And this is why when you look at that border genome-wide, you also don't see an effect, because locally this is maintained, but what you really seem to have is something that's like kind of a bit farther away.
Okay, thank you.
Any other questions from the audience?
Can I ask another?
I had one actually, which was you showed this you showed the heat map showing some changes in differential contacts, and then you showed the change in the saddle plots. I was curious if you looked at it looked like a lot of the changes were happening near the diagonal at close distances. So I was curious if you had looked into the compartmental interactions as a function of distance.
Yeah, I didn't see something obvious. I think what's happening is that in compartment A, like around active genes. Sorry, I'm gonna try to find this. I think what's happening is that at compartment A around active genes is where you see this effect, which is kind of closer to the diagonal. But then when you look at compartments, what you see is this effect on kind of longer range interactions, if that makes sense. Because yeah, I tried to do kind of PFS curves and things like this, and I didn't see anything like striking if you look at A or B compartments or genome-wide.
Got it. Thank you. So let's thank our speaker and thank all of our speakers for this session.